Hi there, and welcome to Miss Elliott's Biology. Today we're going to be talking about proteins and how we make them. All right, so first we need to talk about what proteins actually do. You're probably familiar with proteins being important in hair and muscles, but proteins can also be hormones or messengers within the body. They can also be transporters that allow things in and out of our cells, and they can also very importantly be enzymes. And enzymes are the workers in our cells. They build things up, they break things down, so really important in metabolism and in helping our cells do work. So what do proteins look like is the next question we need to ask. And this is a protein over here. And if we were to take that and to stretch it out into just one long strand, we would see that proteins actually have repeating units of what's called amino acids. So amino acids, they each have similar parts and then one different part. So I'm going to show you that over here. So each amino acid has a carboxyl group, which is right here. They also have an amino group, which is right here. And then lastly, they have a variable group, which we call the R group, which would be the different parts along the bottom, which you see here. So some of the amino acids, because of their different variable parts, some are negative in charge, some are positive in charge, and then others can be non-polar, meaning they don't like water, and some are polar, meaning they do like water. So the positives and negatives, they are going to attract each other, and that's how we're going to get this final kind of shape of the protein in the end. And we'll talk about folding in a little bit. But what's really important about amino acids, they have the carboxyl group and the amine group, and when the carboxyl group of one amino acid meets the amino group of another acid, they form bonds. So here's a bond between those two groups of two amino acids. Here's another one, and those bonds are particular. They are called peptide bonds. They're strong covalent bonds, but they're called particularly peptide bonds, and they hold the amino acids together. So a long chain of amino acids, because they have a lot of peptide bonds, is called a polypeptide or polypeptide chain. So the next thing we need to look at is how proteins are made. And proteins are made in basically a two-step process. Really, a polypeptide is made in a two-step process. The first is in the nucleus, and the second is outside in the cytoplasm where the ribosomes are located. So let's take a look at the first process, which is in the nucleus, and that is called transcription. And the reason it's called transcription is because we're writing the language of the DNA into a different language. So we're actually rewriting it into mRNA. So we're making a messenger RNA copy, or mRNA. And the messenger RNA is just a different kind of nucleic acid that is going to hold the information of the DNA and bring it outside of the nucleus, because the DNA can't go outside the nucleus itself. So here is a gene, and this gene we are going to express. And the way that we are going to make a protein from that gene is first making the mRNA strand, and we are going to use RNA polymerase. Remember, I said enzymes are the workers. Well, RNA polymerase is an enzyme that is going to actually make that mRNA copy. So what you can see it doing here is you can see it opening up the DNA and adding mRNA nucleotides that are complementary to this DNA strand. So it's going to be a complementary strand of the DNA, and therefore it's going to hold the message. So once we have that message in mRNA, we're going to have the mRNA leave the nucleus and find these things that are assembling in the video. So we have the mRNA already. We have the mRNA. It's right here, mRNA. tRNA with amino acids. The tRNA is the T-looking molecule, and the amino acids are going to be the blue circles that are held by the tRNA molecules at first. All right, and then the ribosome is this big purple molecule. It's actually pretty small compared to the rest of the cell, but that ribosome is going to be where the protein is going to be assembled. So you can see on the mRNA, you have triplets, or in other words, three bases, let's call them bases, equals one codon. So a codon is going to attract a certain tRNA molecule, and that tRNA molecule is going to put an amino acid in place in the sequence of our polypeptide. The next codon is going to attract a different tRNA molecule and bring a different amino acid. So our next codon is UAC. It's going to attract a tRNA molecule that's complementary to it, AUG. When those are complementary, it brings that certain amino acid into place. We have a peptide bond formed between the previous amino acid and the next one. And then what happens is our ribosome moves along the mRNA. The mRNA is kind of pulled through it, and as that happens, we get a new tRNA molecule that meets up with that next codon. And this keeps happening until we start to form this polypeptide. And so now we have a chain of amino acids in a particular sequence based on this mRNA, and this last codon is called a stop codon. When a stop codon is reached, no amino acid is put into place, and that signifies the end of the polypeptide. And so there we've made a polypeptide through translation. All right, so after we've made a polypeptide, we need to start folding it. So the first kind of structure, first level of structure of a protein is just the sequence of amino acids that we made through translation. So this polypeptide has a certain sequence of amino acids, and that's its primary structure. Once we start to fold it a little bit in secondary structure is when we get these helices. So the helix, the sheets, different structures that just have different amino acids that are close by interacting and forming hydrogen bonds with one another. The next part of structure, you can see this amino acid, this polypeptide, is moving until it finds its proper shape. So that's its final folding shape. And so all the helices and all the sheets, they are all folded on top of each other, and we get this tertiary structure. Tertiary just means third structure, so T for third. So tertiary structure is that third level that we get that actually has all of the beta-pleated sheets and all of the helices that are twisted on top of each other. So one polypeptide completely folded. However, we probably aren't done yet. Most proteins actually are several polypeptides that are together. So if we have our red polypeptide that we got here, we also have maybe a yellow one, maybe a blue one, maybe a green one, and now we get a protein that can actually do a job. And its structure and its shape is really important. So why is shape important? And the best example of a protein shape being important is we see it in enzymes. So enzymes, like I said before, they do all the work of the cells, and they're really important because they build things up and they break things down. So the things that they work with need to somehow fit inside the enzyme. So here we see that the enzyme has a shape on the top that's called an active site. That active site is very important. If it loses its shape, it can't do its work anymore. So the active site's very, very important because the substrate that it works with needs to be able to fit in. So this enzyme substrate complex, you can see that the substrate fits perfectly into the enzyme. This is kind of like a lock and key. So the lock would be like the enzyme and the key would be the substrate. If the key can't fit in, it's not going to unlock that lock. So here's an example on the bottom of the same thing that I was just talking about. The enzyme has an active site. The substrate is going to try to fit in. If it does fit in, what we're going to see is we're going to see an enzyme substrate complex, and that's where the magic happens. That's where the enzyme starts to do its work. When it does its work, and in this case it's breaking something down, what we're going to get is we're going to get products that are going to be released. The enzyme is going to go back to normal and is going to be recycled. So one more time, substrate, We have the substrate enzyme complex. Products are released. The enzyme is recycled. So thanks for watching, and I hope to see you again soon.